Hello, everybody, and thank you for joining us for today's webinar on best practices for rapid reference quality genome assembly. I am your moderator, Brendan Pyle, and I'm excited to introduce Chai Fungtamassan, our genomics expert and bioinformatics scientist from DNA Nexus, who will be giving the presentation today. A few housekeeping before we get started. If you have any questions during the webinar, you can submit them through the chat or Q&A sections on the bottom of your Zoom window, and we'll answer as many as possible in the questions portion at the end of the webinar. Now, without any further ado, I'll hand it over to Chai to kick it off.
My name is Chai. I'm a scientist from DNA Nexus who work on genome assembly. I have unique opportunity to de novo assemble 68 plant and animal genome for the last couple of years. This is quite a big number, and this includes several genomes that you're familiar with, like human, cow, butterfly. But some of these genomes actually are quite unique and complex to assemble. For example, the corn genome has very high repeat content. Some fish genome has partial tetraploid genome. Also, maize genome is gigantic. The wheat genome is gigantic, and also hexaploid. So throughout this work, I accumulate a lot of knowledge and experience, and develop into this best practice that I want to share with all of you today. Hopefully, you find this enjoyable and beneficial. The topic today will include why we assemble genome. Why is it important? What's the definition of reference quality genome? The best practice in doing genome assembly for long-read technology using FALCON and Canu. The scaffolding to improve assembled genome. Our case study in macaque. So first of all, I want to pose up this question. Why are we doing genome assembly? I think everyone who joined this webinar are going to be aware that genome assembly is quite important. But just to reassure that, I would say that genome assembly, which later on going to become reference genome, is a foundation to study genetics. It is true that you can still do good science without reference genome, but having reference genome is a very useful framework for communication. If I tell you that I discovered a gene that implicates in heart disease, the very first question that comes to your mind is going to be, where is this gene located? Located on which chromosome, which arm of chromosome? This is because we live in the post-omic era, and we are so familiar that genome is organized. Gene doesn't exist in isolation. It exists with all the genes in the chromosome. Having reference genome is also useful for us to find functional elements. You can annotate them to discover what are the metabolic and what are the phenotype that you might expect to see in this species. Also, having reference genome can allow us to study about interaction between different elements in the genome. For example, you might try to figure out what are the enhancers that are associated with particular gene. You can take a look based on location and also the interaction with other elements around it as well. Also, if you have the gene on the same chromosome, then you can know that the selective pressure on this gene can be different from the rest of the genome, which is on the autosome. Right now, there are almost 900 reference genome in NCBI. So we usually got a question, why do we still have to assemble more genome? Why can't you just try to align your data into the existing reference genome? I would say that those approach is beneficial, but I would argue that having the correct reference genome for your species gonna be much more accurate. It can give you better result. And also, in some cases, you might already have reference genome for that species, but by doing de novo genome assembly, it can help you discover complex variants, which may not be able to find using the existing reference genome. And finally, having multiple reference genome gonna benefit the community as a whole, because you can use multiple reference genome to do comparative genomics, and also you can perform study like the evolution of how this genome evolved over time. And of course, because I mentioned that reference genome is quite important, so definitely we want to create good genome assembly. Now it comes into the question like, what's a good reference genome look like? What's the requirement to have a good genome assembly? I can use the definition from the VGP Vertebrate Genome Project group. This group is a multinational and this group is a multi-institutional and multinational organization which aim to make the high quality reference genome available for 10,000 of the vertebrate species. We work with them to provide data analysis pipeline on the cloud, and we also learn several best practices from them. Before I discuss about the criteria to consider certain genome to be high quality reference genome, let me give you some background in genome assembly. In the genome assembly process, you start from the read of the DNA sequencing data, and then you try to assemble that into the contiguous sequence of DNA, which is called contig. This process is what people usually refer to when they say genome assembly. If we think of read of DNA just like the jigsaw, the easiest way to figure out where each read belongs is try to align to the reference, which is in this case is a picture on the box of the jigsaw box. But however, in genome assembly, we don't have reference genome available, and basically we want to de novo assembly to create it. We didn't want to rely on what we know about the genome. So what we need to do instead, we have to take each piece of jigsaw and do all against all comparison to figure out which piece should be connected to each other. This operation for genome assembly is quite expensive, and roughly the order of growth is about n squared. So after you assemble genome with the contig, then you can further improve it using other technology. For example, you can use Hi-C, BioNano, or link-rich from 10x genomics to figure out the orientation and the order of this contig. We're gonna call this level of organization as scaffold. And scaffold is the standard representation of the haploid genome. But rather than stop there, we can further improve your genome assembly. To be precise, we can phase it. Phasing is a method that figures out which variant came from the same chromosome or from the homologous chromosome. So what do we get in the end? We get both sets of chromosomes, one from father and one from mother. This seems like buy one, get one free because you do genome assembly once, but you get two genomes out of it. So these are the basic concept and terminology of genome assembly. Now let's come back to the terminology, to the criteria from VGP to consider which one is a good high quality reference genome. The first criteria that VGP poses is that you have to be certain about the base pair that present in your assembled genome. And in this case, they use a cutoff called QV40. Roughly, it requires that the base call error rate has to be less than 1 in 10,000. Second, the assembled genome has to have high contiguity. This index is called N50, and roughly is the weighted median. The way that we calculate the N50 is by sorting your assembled contig by size, and then you calculate the cumulative length of the contig from the largest to the smallest one. In this case, you can see here that we start from the 14 megabase pair, and once you go to the next contig, this is going to be 8 plus 14, which is 26. So in this case, you can see that that length that you calculate is already exceeds half of the assembled genome size. So the N50 of this particular genome assembly is going to be 8 megabase pair. Roughly, the higher the N50, the more contiguity of your assembled genome. The VGP also requires that the scaffold has to add N50 at least 10 megabase pair. More than 90% of the genome has to be able to get assigned to chromosome. It has to be phased, and finally annotate for function element. So now let's play some guessing game. How many vertebrate genome among 900 in NCBI that had meet all these 6 VGP standard? This is actually a tricky question. The actual answer is actually 0. So there are several good genome assembly out there, but they have not been phased yet. But if we drop that criteria for the phasing genome, we actually have 16 out of about 900 reference genome NCBI that meet the rest of the standard. The bottom line is creating this high quality reference genome is not trivial. And more than that, some species is much harder to assemble the genome than the other. In particular, several plant, fish, and amphibian genome are polyploid and have high repeat content, which is very hard to assemble. We cannot go over several techniques today that can alleviate this high repeat content issue. The next part is going to be the main part of my talk today, which I will share my experience about how to create good assembled genome. When we talk about best practice, people usually think of magic parameter that allow you to run this genome assembly software and get the result. But I would argue that the best practice starts from the good experimental design, the good sample selection, the quality control. You can see that there are both components of the experiment and computational involved in creating high quality reference genome. Also, this is not the straightforward process. This has to be iterative sometimes. For example, if you know that from your sample after you choose it, and if you do some sequencing and you found that this sample is not high quality, then you might have to change the sample. And also after you do genome assembly, you might have to do several rounds of the evaluation to figure out and get a better genome. So the very first step in doing genome assembly is to choose a good sample. And I would highly encourage you to choose the inbreed individual. The inbreed individual, which is mean that you have been breed particular group of population for a long time, and the genetic variation in that species in that particular individual is very low, between 2 copies of the diploid. So the inbreed is like the haploid genome because you don't have a lot of variation between 2 copies. This way, you don't have to worry about the heterozygosity, which you're going to require more data to assemble, and also it's going to be more complicated in the assembly graph. In the case that you don't have access to the inbreed individual, I recommend that you take a look at the trio information. For example, if you have, let's say you want to assemble a cow genome, and you know that you have access to the genome of the father and the mother of this individual, you can actually use the data from the father and the mother to figure out which group of the data in the offspring that belong to the paternal side and which group belong to maternal side. And then you can assemble each group separately. As you can see in this publication from Sergey Korin group, they use the approach I mentioned to create very long contiguity and also phase genome using trio approach. Next is about contamination. So I hope this is quite intuitive, and I can have more examples later on. But when I mention contamination here, I didn't mean just to get the sample that doesn't have the DNA from other species. The contamination can come from the same species as well. For example, if you want to assemble the plant genome, plants usually have a lot of chloroplast genome, and you might want to get rid of those chloroplast genome first. Otherwise, your data are going to be highly contain the chloroplast genome, which you don't want to assemble. And finally, make sure that you have enough DNA sample. This can be tricky for the very small insect because it's very hard for you to get a clean sample. So you might have to pull several samples together, and the problem of the heterozygosity is going to get worse. But in general, if you can avoid that, try to get a clean sample and make sure that you have spare material enough for more experiment. It is possible that you have to do more round of the DNA sequencing, and also having additional material going to be helpful for you to run a scaffolding tool on the sample later on. Here's some case study about contamination. So when we start genome assembly, the very first time that we do, usually we get very good results. So we just get the data and we just assemble right away. But we run into this corner case that we get the insect sample from the collaborator, and when we assemble it, we get nothing back of the program, and we curious why. So we take a look at the raw data, and we found that if we blast the raw data of this insect the DNA to the NCBI, we found that they actually map to microbe data, not the insect. So this means that this sample is highly contaminated, and we have to talk with our collaborator to change the DNA sample and provide some new data. And later, we can create good genome assembly from that later on. So after we run through that scenario, now we have the standard protocol that every time we got data from collaborator, we have to check for contamination first. So in this case, I got the plant genome data, and after I checked on the NCBI, I found that most of the data actually map to plant as we expect, but about 20% of data map to fish. So we discussed with collaborator that this data is contaminated. But then they suggest that maybe we can just try to assemble and see what happens. Hopefully we can get some contig that belong to plant, some contig that belong to fish. But unfortunately it's not. So we got a lot of contig that contain both plant and fish genome in the same contig. So this assembled genome is not very useful in the end. The bottom line is, if you try to check for contamination first, if you found that your data is contaminated, try to get rid of the contamination part to make the data clean before running genome assembly. The next part is on the sequencing and QC. So the message that I want to point out is that when you plan your genome assembly, try to decide the library that's gonna give you the long subread. Right now, from what I see, most data that we get from customer has the N50 of the subread of around 17 kilobase pair. Sometimes we still get the data of 10 to 12 kilobase pair, and we can still get good results from them. However, overall, inevitably, the longer the data, the better the result. Especially if you work with a species that has high repeat content, I highly encourage that you try to aim for even longer N50 of the subread data. For example, 20 kilobase pair. So why is the long read is important for the high repeat content species? If you remember the jigsaw analogy that I mentioned earlier, the repeat content is just like this blue area of the sky here that you can barely assemble them, because each of the jigsaw piece here look very, very similar. So that's the same as repeat content. Because your read is quite short compared to the repeat region here, you can actually make sense, you can actually assemble into the full picture here. And in the end, you're gonna have the genome that is quite incomplete. So some people sometimes say that you can just skip the repeat region because it's not that important. But actually a lot of function is harbored inside this repeat region. For example, Fragile X syndrome or Huntington disease. Those are all related to repeat region. However, if we have the long read data much longer, we can actually cross over those repeat region and create more complete genome. So the longer the read, the better the genome assembly in general. After you make sure that you can have the data that long enough, then it's time for you to think about getting sufficient coverage of the data. So the current recommended value is to get 50x coverage per haploid. For example, if you have the diploid species that is highly homogeneous and highly homozygosity, you can just use 50x coverage. That should be sufficient. But if you work on a species that has very high heterozygosity, then you might want to get 100x coverage. Also, it's important for you to estimate the genome size from your sample first before you run genome assembly, because the genome size and the repeat content, it's very important. They're gonna be very important in the parameter setting later on. Usually we can get this estimated genome size and repeat content from literature, but in the case that you don't have access to that, you can try to use data from some wet lab experiment. For example, you can try to run flow cytometry and figure out what is the genome size for your species. Also, if you have Illumina data from your sample, you can estimate the kernel frequency and then use tool like Genome Scope to interpret kernel frequency into genome size and repeat content. Even though I mentioned several times that you try to get long read data and also high coverage data, but it doesn't mean that you should overload your flow cell with your data, because if you do that, the data that come up with is not reliable and cannot be really used. And finally, if you have the project to assemble very large genome, make sure that you don't do them all at once. I recommend that you try to take small amount of sample, try to sequence it first, and then make sure that your library has sufficient subread and also lack of contamination. And after you make sure that this data is clean and good, then you can go full scale to create all the data. The next part gonna be on about running genome assembly software. So there are several tools that can do genome assembly from long read data. Today, I'm gonna focus on two family group of tools. The first one is a FALCON. The FALCON is a tool developed by Pacific Bioscience company, And this tool is a Diploid-Aware hierarchy genome assembler. Roughly, it takes the long read data and then perform all against all comparison to create the error correction read. This error corrected read can be later on assembled into the assembled genome. This approach will found to create much more contiguity than using the short read assembler. After you run Falcon, you can also use FALCON-Unzip to fetch your genome. Roughly, this FALCON-Unzip can take data from your Falcon and then figure out the part that each read contribute to the final assembly. And after that, it can provide you the primary contig, which represent your haplotype genome. The haploid genome. And then on the region that have sufficient information to separate out the haplotype, you're gonna get the haplotype contig as well. Internally, we also perform the benchmark to test FALCON-Unzip on human data. And we found that in most cases, those contigs are quite accurate. There's no single base pair of the misfit SNP on it. Most of the error when it happen also quite random rather than a misjoin two contig together. After the FALCON-Unzip, you can solve out one called Falcon phase. Falcon phase is a collaboration between Pacific Biosci and Phase Genomics. Roughly, it uses high-C technology to figure out which are the haplotypes that came from the same parents. And the end product of that is the full phase haplotype genome. There's another series of the tool that's called Canu, developed by the developer group from NGHRI. Canu tool is derived from cellular assembler, and it can work both with PacBio and Oxford nanopore data. As I mentioned earlier, that this assembler has to do all against all alignment, and this is a quite compute-intensive protocol. As you can see here, when we go from the min genome to human genome, even though the genome size increased about 6 times, the total core hour increased much more than that, about 8 times. And when we go from human to the wheat genome, you can see that the required core hour increased much more. If we interpret that into human time, you can see that the waiting time for the wheat genome assembly is more than human lifetime. Ultimately, you have to find a powerful computing resource when you do genome assembly, and you want to run things in parallel to get results in reasonable time. Once you have secured your computing resource, here's what I recommend for running genome assembly. First of all, when you start, I recommend that you start with the parameter from the species that's similar to your species first. In the case of the Falcon, you can find those parameters from literature or from the collection of parameter set released by the PacBio company. In the case of Canu gonna try to choose those parameters for yourself, for you, so you don't have to worry about it. But that's not always true. For example, if you work with high repeat genome, you're gonna have to put a hard cap on the glimmer frequency and a large minimum overlap requirement for the Falcon. And on Canu, that's gonna be the overall more threshold parameter. The idea is that if you allow too many alignment from the high repeat genome, most of that alignment gonna come from repeat region, and it's not gonna be useful for your genome assembly. Also, you might consider this tool released by the Gene-Meyer group. This tool is called 10-mask and RepMask, and the rough idea is try to mask the repeat content in the raw data first before genome assembly. But I had to tell that from our experience, this tool, in most cases, it worked very well. But there are some corner cases. For example, in the maize genome, there's a very high repeat content, and you mask your data using this RepMask and 10-mask first. You can barely create any good genome assembly from that. And finally, I usually get a question like, should you do Falcon or Canu? I would say that if you want to do follow up by FALCON-Unzip on Falcon phase, definitely you have to go with Falcon. If you have Oxford nanopore data, then you have to use Canu. But if you have data that work for both too, I recommend you to experiment with them. And also, technically, you can combine them. For example, you can run error correction with Falcon and then assembly with Canu. And finally, on the evaluation part, this part is, even though I put it to the end of the pipeline, but technically you might have to run several rounds of assembly and then evaluate to pick the best one before you proceed with downstream processing. The very first parameter that people usually take a look in genome assembly is the N50. But I recommend that N50 is quite vulnerable to the assembled genome size. For example, if your genome is supposed to be 3 gigabase, but your assembled genome base pair turned out to be like 6 megabase, you will know that the N50 here that you got can quite inflate. So rather than using N50, I recommend that you consider NG50, which is normalized rather than by the total assembled base, but by the expected genome size instead. With the NG50, the result is going to be more comparable between different experiments. And besides the read length distribution, I recommend that you take a look at the expected gene that you should find in that species. In this case, I recommend you use BOOSTCO score. The BOOST score cannot report you the percentage of the expected single copy gene in your particular species. You may also consider the orthogonal technology to evaluate it. For example, you can use BioNano, which is going to create the optical mapping. And then after that, you can use hybrid scaffolding to check the conflict between your assembled genome and BioNano. And this misjoint conflict report here is a good indicator to tell whether your assembled genome is correct or not. Finally, if you're concerned about the 5-scale error rate, then you can try to use Illumina data mapped to your assembled genome and estimate mismatch percentage as well. You can do more experimentation with your assembled genome as well. For example, you can try to align your assembled genome to the closest genome species. I recommend this tool developed by Maria Novastad from our company. This is the interactive plot. It is open source, so I highly recommend you to visit it afterward. And then after you use this criteria and make sure that you got the best assembled genome ever, don't forget to publish them, because if you didn't publish using the PacBio data, the assembled genome has high error rate, and I recommend you to fix them using the publishing tool first. The next part is going to be on the scaffolding and improve your assembled genome. So the method to do scaffolding, I recommend you to follow this protocol from the VGP group. Roughly, after you run the genome assembly by FALCON or FALCON-Unzip, then you can scaffold it using Scaf10x to combine 10x data with your genome assembly data to create a scaffold. And then you do similar thing using data from the BioNano and the Hi-C as well. And after that, you can try to evaluate the correctness of your assembled genome. Try to close the gap using PBGLE, publish using PacBio data using Arrow, and finally use Freeway or Pylon to publish your data using, to publish your assembled genome using Illumina data. Now I want to give you more real example. So here's a case study from La Jolla Institute of Allergy and Immunology at University of Louisville. They are interested in macaque genome. So macaque genome is actually not hard to assemble, but what this research group interests is in immune recognition region, which is full of segmental duplication and have very high variation among individuals. So what should we do in this case? First, we communicate with them to make sure that the N50 of their raw data is long enough. This long N50 data is very essential to capture the segmental duplication. When we get the initial data, the initial data has an N50 of around 9,000 base pair. But then we communicate with them that it's insufficient, so they were able to increase that into 16,000. We run this assembly in the cloud, so it takes 4 days. Rather than try to run this on the single core machine, it's going to take about 5 years. And after that, we found that the result come out pretty good. First of all, you can see here that the contiguity of both primary and haplotype contig come out very good. The N50 and NG50 is in the level of the 7 to 8 million base pair. And then so the haplotype contig come out pretty long as well. The second thing is that you can see here that the haplotype contig in terms of number of base pair here is almost the same as the primary contig, which means that this genome actually is quite heterozygous, and it's quite the right choice that we choose FALCON-Unzip. So we can get a lot of information from the other haplotype that present in the genome. So we gain a lot of experience through this collaboration, and we have several consortium to do great science, including BGP. You can learn more about what we do and what we offer as a genome assembly service from this URL below. Today, I hope I convinced you that genome assembly is very important. We discussed the definition of high-quality reference genome from the BGP group. We discussed about best practice in genome assembly and scaffolding. Several case studies. I would like to thank Brett Hannigan, who was instrumental in setting up most of this pipeline on the Nexus and make all this work possible. Thanks Maria Sambaski for update this tool to be in good condition. Thanks Jason and the whole science team for several recommendations in parameter setting. Also, thanks everyone in marketing team and research group for helping with this webinar. With that, I would love to take any question about genome assembly. Thank you.
All right, thank you so much, Chai, for that presentation. Just a reminder, everybody, if you want to submit questions, you can submit questions through the Q&A icon or the chat icon at the bottom of your screen. And to answer one question real quick that came in, are the slides available online? The slides will be made available in a follow-up email about a day or so afterwards, so just keep an eye on your inbox and we can send those over to you. All right. Now, our first question for you, Chai, here is, since both PacBio has high sequencing error, is it effective to test for the contamination on raw data rather than after error correction?
So the program of choice here we use BLAST, and BLAST actually allow a wide range of the dissimilarity between your data and database. For example, you can still align human to the E. coli data, which is quite different. With that, I would say that the raw data itself is sufficient because the error is just about 15%. So you can still easily match your raw data to the data in the NCBI database.
Okay, perfect. Got a question here about the N50 read length. They're asking if you can explain a bit more about that. Since the N50 is the minimum contact length needed to cover 50% of the genome, is it shorter?
So the N50, the rough idea is that if you saw the genome by size, right, and then you calculate the cumulative length from the largest county to the smaller one. So once the total accumulated size is above the total assembled size here, you can see here that the total assembled size is, let's see, this is 10, it's 20, about 30 megabase pair, right? So if you combine 14 with 28, they're already more than half. So the N50 of, in this case, going to be 8 megabase pair.
Perfect, thank you, Chai. Our next question here is, what is the computing requirement required for scaffolding?
So I would say it quite vary between on the stage of the scaffolding. The Scaffolding and the bio-nano hybrid or the high C, they are not compute intensive. But there are some scaffolding steps that are quite compute intensive. For example, before you can run the hybrid scaffolding here, you have to run the bio-nano assembly first, and that is compute intensive. And also the gap closing by visually also very expensive as well.
Okay, perfect. Our next question is asking if you can just explain a bit more about running Falcon and Canu.
All right, sure. So in the Falcon genome assembly, first of all, you provide the program with the package file or the BAM file, right? And then the very first step is that it cannot convert your raw data into the FASTA file. And once you get the FASTA file of the raw data here, it will attempt to perform the OR against our alignment, and the aligner program is a de-aligner. So this process, roughly, it tries to figure out which read should be grouped together and correct for those errors. So after that error correction, you're going to get the error corrected read, which then just assemble again into the full assembled genome here. And then I mentioned earlier that there are downstream steps that you have to polish the genome. So in that case, you're going to have to take the the package file or the BAM file to correct for the error that present in your assembled genome again. As for the Canu is a little bit different in terms of the algorithm, and you can see here that it requires 3 steps rather than 2 steps. In case of the Canu, you just provide the fast A data, and then it can perform the error correction, the trimming, and the assembly. And the experience from using Canu is going to be quite different because you just keep it, and then by default you don't have to provide any parameter at all. It will just figure it out by itself, and then you get the assembled quantity from it. But then, as I mentioned, that it doesn't necessarily mean that you never have to modify the parameter, because in some case, like high repeat genome, you still have to modify this parameter.
All right, we are receiving a couple questions about specifically Illumina data. So the first one is asking if there's any Assemblies solution for Illumina data on DNA Nexus.
We have several tools for the short-read assembler. We have SSAKE. We have, I think we have Velvet. So the short-read assembler, it's gonna be a lot easier to operate. So I didn't do it as a professional service. I let customer run it by themselves.
All right, perfect. And then our next question on Illumina data is wondering if it's possible to make a hybrid assembly using PacBio and Illumina data, and then if so, what software?
There are two out there that can do this assembly actually. I recall 2 of them. One is Marvel, and Marvel is the one that used for the salamander genome. I think the actual name is Sotel. It would hard to pronounce. And then another program is Mazuka. So both programs can do the hybrid assembler. Right now, we don't have these 2 programs yet because we didn't have anyone request for it yet. But if you have this data and you are interested to do hybrid assembler, let us know afterwards so we can consider put them on the platform.
All right, perfect. Thank you. We've got one more question here. How much coverage is required to assemble perfect PacBio reads without using Illumina short read polishing?
I would say that the recommended coverage is depending on your heterozygosity. Let's say if you work with the human genome, I would say 50x is fine. But you have very high heterozygosity, then you're gonna need like 100x coverage for that. But with that said, it also depends on your N50 overall data as well. Because if your read is very long, then 50x is fine. But if your read is very, very short, then I recommend you to get a little bit more to compensate for that. But overall, having long read is more important than getting high coverage data. Internally, we actually perform some benchmark and try to see what's the improvement from having Illumina data in your in the whole process as well. And for what we use Illumina so far is for another route polishing using Pylon or the for the freeway, but we didn't use them directly in the genome assembly process. So in terms of contiguity, it's not going to change, but you can get better file scale error. Basically, the error rate per base pair going to be lower if you do another route of polishing using Illumina data.
All right, perfect. Thank you, Chai. I think that will do it for us for today. Just a reminder for everybody, we'll be sending a follow-up email in the next day or so. That'll have a link to the recording and to the slides as well. So I just want to thank everybody for joining us and have a great rest of your day.